So, welcome to this class on neuroscience of human movement. We are continuing with our discussion on cerebellum. So, this is part 2 of our discussion on cerebellum. So, in this class we will be talking about cerebellar cortex, different from cerebral cortex. So, this is cerebellar cortex and the different layers of cerebellar cortex and the types of inputs that are received by the cerebellum and the type of output in the cerebellum. Okay. So, we said that the cerebellum is located inferior and posterior to the brainstem and inferior to the cortex. So, if you take a particular area, a relatively small area of its cortex or the cerebellar cortex is going to look, it has this unique 3 layered structure. So, there is one layer there and a relatively thin layer there and a thicker layer at the bottom. So, these layers are called as M, the thinner layer is called as P and the deepest layer is called as G. What are these? It is not miles per gallon, but these are rather molecular layer, Purkinje layer and the granular layer. Okay. So, these are the 3 layers of the cerebellum and of these, so a bit bigger picture is in this slide. This is relatively confusing, but we will take step by step to understand what is going on. So, the inputs come from the bottom. So, this is white matter. So, at the granular layer which starts around here, right, this layer is the major input layer of the cerebellum. This is a major input layer of the cerebellum. What this has is a large number of granule cells. What is this number? We mentioned in the previous class, this is about 100 billion granule cells. That is a lot of cells. 100 billion, approximately 100 billion granule cells are there in the granular layer. So, they are called as this layer is called as granular layer because it contains a large number of granule cells. The next layer is called as the Purkinje cell layer. It contains cell bodies shown in purple here, these cell bodies of the unique and distinct cells that are cells that are unique and distinct to the cerebellum. These cells are called as Purkinje cells. They are also highlighted elsewhere, for example, their cell bodies are located in the Purkinje layer or Purkinje cell layer. So, what does that mean? When I say that their cell bodies are located in the Purkinje cell layer, it means that their dendrites and axons are elsewhere. Their dendrites extend into the molecular layer. Actually, they have too many dendrites not shown in this picture. Each Purkinje cell can have dendrites from anywhere from 100,000 to about 250,000 dendrites. That is a large number of dendrites. So, each Purkinje cell has about 100,000 to 250,000 dendrites, and these dendrites are in the layer above the Purkinje cell layer or the molecular layer, and the axons of these Purkinje cells project onto nuclei embedded within the cerebellar cortex. These nuclei are called as the deep cerebellar nuclei. So, these are the major output structure of the cerebellum. So, the Purkinje cell layer in some sense can be considered to be an output layer. In the molecular layer, what you have are dendrites of the Purkinje cells, and it turns out that the axons of the granule cells, so the granule cells we said are a very large number. I am going to be throwing out such large numbers, so please be cautious in interpreting them, and please verify, read them and understand and make sure that you get the numbers correct. These granule cells, their axons ascend upwards and then proceed perpendicular to the dendrites of the Purkinje cells. But then we have 100 billion granule cells. It turns out that these granule cell axons, when they go, and there are several of these that go like this, right, so from each Purkinje cell, they all go parallel to each other, but perpendicular to the dendritic tree of Purkinje cells, right. Because they are parallel to each other, these are called as parallel fibers. For illustration, only a few of these are shown. Obviously, what is there is a way more complicated picture, but what we are showing here is only a few, only like few tens or few less than 10 parallel fibers or a little more than 10 Purkinje cells. What is actually happening is that there are 100 billion granule cells, and each Purkinje cell is innervated anywhere between 200,000 parallel fibers to 1 million parallel fibers. So, one Purkinje cell receives input from 200,000 to 1 million parallel fibers. That is a large number. So, all these are not shown for illustration purpose, only we are showing a small number of these connections. It turns out that this interaction between the input layer or the granule cells and the output layer, which is the which is arguably that is the Purkinje cell layer, happens at the molecular layer. So, this is the major processing layer of the cerebellum. So, what is going on? Let us remind ourselves one more time. So, what is going on is that the major inputs are received at the granule cell level, at the granule cell layer or the granular layer. This input proceeds further to other layers via the granule cells and its parallel fibers. The Purkinje cell layer contains the cell bodies of the Purkinje cells, whereas their axons project to the deep nuclei, and their dendrites are in the molecular layer, and these dendrites are contacted by hundreds of thousands of parallel fibers, hundreds of thousands of parallel fibers. So, the molecular cell layer then or the molecular layer is the major processing layer of the cerebellum. Purkinje cell layer is the major output layer of the cerebellum, whereas the granular layer is the input layer, right. So, this what is not discussed is that the cerebellum also contains other cells such as basket cells, an interneuron, an inhibitory interneuron shown here, that is the, I am going to write this here for clarity, that is the Golgi cell, and a different type of interneurons here. These are the interneurons, the star shaped interneurons. So, they are called as stellate cells. What is not shown in this picture are other type of cells. These are Chandelier cells, Lugaro cells, and unipolar brush cells. There are several other cell types, but for the purpose of this course and discussion, it is enough if we understand the functions of the granule cell the Golgi cell, the Purkinje cell, basket cell and stellate cell for the purpose of this class, because otherwise the class becomes too detailed. It turns out that the granule cells receives inputs from one of the major sources of input to the cerebellum. These are called as mossy fibers. Okay. These mossy fibers serve as input to the granule cells. So, each granule cell receives input from a few mossy fibers, but one mossy fiber innervates several thousand granule cells. That must be true, right, because it turns out that the total number of granule cells is 100 billion, and that means, and also we said earlier in the previous class that that is greater than the sum total of all the neurons in the central nervous system and the rest of the cerebellum, right. So, that is more than 50% of the neurons are in the granule cell layer, are composed of basically the granule cells, is it not. So, each mossy fiber then innervates thousands of granule cells, but one given granule cell receives input only from a few mossy fibers, right. So, these, what are these mossy fibers? We say mossy fibers. What are these? These are sensory information from the spinal cord and other information from the cerebral cortex. So, for clarity, if I say cortex now it is confusing whether it is cerebellar cortex or whether it is cerebral cortex. Mossy fibers are input neurons that arise from spinal cord and other input regions of the cerebellum and from the cerebral cortex through the pontine nuclei. Okay. So, mainly from these two sources, and maybe from other sources also, from, for example, from the vestibular system, for example, from the visual system, from other systems also, but mainly from the cortex and the spinal cord, for example. So, that is sensory information and other information that comes from the cortex. These are called as these cells that bring this information into the cerebellum are called as mossy fibers. And then there is a special fiber that arises from the inferior olive, okay, that arises from the inferior olive. These type of fibers are called as climbing fibers because these fibers climb through the granular cell layer. They do not project to the granule cells, but rather they directly control the action of the Purkinje cells by connecting or by innervating, by making synapses with multiple dendrites of Purkinje cells, like it has been shown in this picture. So, they climb over the Purkinje cell body, cell dendrites, and they make multiple connections, and they do not project to the granule cells. So, whereas the mossy fibers project to the granule cells and through the granule cells control or modulate the function of the Purkinje cells. So, let us remember the differences. So, mossy fibers are indirect excitation, they provide, mossy fibers provide indirect excitation to the Purkinje cells, whereas climbing fibers provide direct and relatively large excitation of the Purkinje cells. Sorry. Okay. So, we will discuss the details in future classes, but it suffices to know for now that the climbing fiber directly and in a greater manner influences the function of Purkinje cells, whereas the mossy fibers first synapse with the granule cells. Actually, they also synapse with the deep cerebellar nuclei not shown in this picture. So, the mossy fibers synapse with the granule cells and these granule cells take information. After they become parallel fibers, they innervate multiple Purkinje cells and modulate action of Purkinje cells in an indirect and less with less amplitude when compared with climbing fibers. So, fundamentally different mechanisms. So, we will continue this discussion, or this theme will continue throughout our discussion of cerebellum. Here is another picture showing one more time the Purkinje cells, Golgi cells, granule cells. So, this is a granule cell, and these are, this is a Golgi cell, this is a granule cell, and you see the distinct tree like structure of the Purkinje cell when compared with the other cells, and how climbing fibers climb, and there are different types of Golgi cells, Golgi cell type 1, type 2, and then there are basket cells, right. These are the various cells within the cerebellar cortex. Some of them are not shown, but many of them are shown. So, one more time, here is the view of the Purkinje cells, right. So, their cell body is in the Purkinje layer, this is in the Purkinje layer, their axon is going to the granular layer, the granule cell layer, and their dendrites are in the molecular layer. Look at the dendritic tree structure. This is a relatively small Purkinje cell with a small number of dendrites. I said that or at least a smaller representation for illustration purposes. We said that this has a relatively large number of dendrites, anywhere between 100,000 to 250,000 dendrites. An important point to note is that the Purkinje cells are thick, or one would think when you are viewing that this is like a tree, you would think that this is a 3D structure, just like a real banyan tree, for example. This is not true. What is actually true is that this Purkinje cells occupy a relatively large territory in the anterior posterior direction. This is the AP direction. They occupy a relatively large territory in the anterior posterior direction, but when you are looking at it from the mediolateral direction, when you are looking at it from that direction, right, it appears to you as if it is very thin. For example, the paper, for example, is actually a 3D object, right, except that its thickness is much smaller when compared with its length and breadth, as one would think. So, there is that is the length, that is the breadth, but its thickness is very small. It is actually a 3D object, but for most practical purposes it can be considered to be a 2D object, is it not? So, for most analysis we could consider that. Likewise, the Purkinje cell is actually very thick in the anterior posterior direction, but thin, very thin in the mediolateral direction, and this is important for their function. Why? We will see in the next class. Okay. And an important concept is the concept of glomerulus. So, we said that there are Golgi cells, granule cells, and mossy fibers. The mossy fiber axon, or the bulbous axon of the mossy fiber, makes connections with the dendrites of the granule cells. Yeah. So, these are the various dendrites from multiple granule cells, right. For simplicity, only 2 or 3 granule cells are shown. Actually, we said that the number of granule cells contacted by each mossy fiber can be of the order of several thousand, right. So, and the mossy fiber also connects with the axon of the Golgi cells. So, thus exciting the Golgi cells. What is the function of this Golgi cell? We will see in the next class. So, these Golgi cells also are innervated by the mossy fibers. Note the number of mitochondria here, the large number of mitochondria here. There must be very critical or very high metabolic activity of this massy granule cell Golgi cell connection area are what is called as a glomerulus. Okay. So there must be a high metabolic activity that must be happening at the glomerulus. That is probably the reason why there is great distribution of mitochondria in this region, right. So in summary, what we have seen is that so the cerebellar cortex is composed of 3 layers. We said these are the molecular layer, Purkinje cell layer, and the granular cell layer, and we described the types of cells in each of this. So we said that the granular cell layer contains mainly the granule cell and some Golgi cells. The Purkinje cell layer contains the cell body of the Purkinje cells, whereas the molecular layer contains inhibitory interneurons such as stellate cells, basket cells, and there are other cells also such as Chandelier cells, Lugaro cells, unipolar brush cells, etc. Right. So we discussed the cerebellar cortex and the 3 layers of the cerebellar cortex. What are these? This is molecular, Purkinje, and granular. This is the new MPG, right. Molecular, Purkinje, and granular cells, granular cell layers are the 3 layers of the cerebellar cortex. And then we said there are different types of inputs and into the cerebellum. These are massy fibers and climbing fibers, and the Purkinje cells are the major outputs, or at least modulate the outputs of the cerebellum, or basically they project onto their axons of the Purkinje cell project onto the deep cerebellar nuclei. Where are these deep cerebellar nuclei? We have not discussed that, we have not shown that in this picture. Actually the deep cerebellar nuclei are embedded within the cerebellar cortex. These are relatively dense structures that serve as independent nuclei to output information from the cerebellum. These are embedded within the cerebellar cortex, but was not shown in the picture that was discussed. We will discuss that and other things in future classes. So coming up next, in the next class we will be discussing about the microcircuitry involved within the cerebellum. So thank you very much.